Hello there. Welcome back to Dr. K. Prem-Primer lecture series presenting by Dr. K. Prem Sagar. That's me. Today we will talk about exonucleases. Exonucleases. So exonucleases are the one of the category of nucleases. What is meaning by nucleases? Nucleases means enzymes which catalyzes the breakdown of phosphodiester bond of nucleic acid. So the phosphodiester bond of nucleic acid is broken down by enzymes. They are called as nucleases. Means nucleases catalyzes the breakdown of phosphodiester bond of nucleic acid. Those enzymes are called nucleases. Nucleases are two types. One is endonuclease and exonuclease. Based on their mode of action, based on their mode of action, where they act within the nucleic acid or outside the nucleic acid or terminus. So based on where their activity is initiated, based on activity initiated, location of the where in the nucleic acid, wherever they start or wherever they initiate their activity, based on that, the exonucleases are categorized into two classes. One is endonucleases and the second one is exonucleases. Endonucleases means they cut along the nucleic acids within DNA, right? Or RNA. They are endonucleases. Best examples are DNases, S1 nucleases, and famously restriction endonucleases. Then today's topic is the second class, that is exonucleases. Exonucleases. Exo means at terminus or at exit or at end. Exonucleases act on nucleic acid molecule at the end or nick or at the terminus of nucleic acids. That's why these nucleases are called as exonucleases. So this is what is today's our topic. So try to subscribe my channel for many updates, right? So as I told you that exonucleases catalyzes the or initiate the activity from the ends or terminus or nicks of the nucleic acid molecule. Yeah, here I have taken one small fragment of double strand DNA molecule, right? You can see here each nucleotide is connected and this is a phosphodiester bond, right? So exonucleases, you see, I told you that exonucleases acts at the ends or terminus or at the nick of nucleic acid molecules, right? Means their activity is initiated at the terminus or nicks or at the ends. This is what is a one double strand DNA molecule. So you see, this is a terminus, 3' end and 5' end, and this is a 5' end, this is a 3' end. In addition to this one, you see, so enzyme can act from a 5' or from the 3'. Both are at their terminus, right? Then here you see, this is what is a nick. Nick means single strand break is called as nick. In the double strand, only one strand is phosphodiester bond is broken. That is called as a nick. So even exonuclease can act at the nicks of the nucleic acids, right? And one more nick is here. You see, T and C. This is what is nick. So it can act from the nicks of the nucleic acids also. The activity initiated from the nicks or terminus or ends of the nucleic acid. Hence, those nucleases are called as exonucleases. And one of the special feature of these exonucleases is they act one nucleotide at a time, means this, they will degrade the one nucleotide at a time from the ends or the nicks. This is what I'm going to explain with this cartoon here, right? And you see, and you this exonuclease can start from terminus or from the nicks. This is the nick, again nick, and terminus or ends. So I told you that exonucleases act from the nicks or ends or terminus, but only one special thing is that it will act one nucleotide at a time. That's what I want to illustrate with this figure. So this is a double strand DNA molecule, and this is a new exonuclease, right? So you see, it is passing through that. It can from 3' to 5' direction or 5' to 3' direction, and you see one nucleotide, again one nucleotide, again one nucleotide. See, this is what is the exonuclease special feature. You see, at the end of reaction, you'll have mononucleotides. Mononucleotides, right? Understood? Yeah, now we will go for a definition of the exonuclease here. Active at the ends of nucleic acids and cleaves one at a time from the ends, either 5' or 3' ends. So active at the ends of the nucleic acids and cleaves the one at a time from the ends, either 5' direction, 5' terminus or 3' terminus. Hope you understood. Next we will go to the uh, types of exonuclease. So nucleases, types of nucleases. Here, exonucleases also classified again based on the activity, orientation of their activity. So they are classified into two categories. The first one is 3' to 5' exonucleases, or 5' to 3' exonucleases. The second category is 3' to 5' exonucleases. So whether they start from 5' terminus or 3' terminus. So based on that, exonucleases are again classified into two categories. They are 5' to 3' exonucleases and 3' to 5' exonucleases. And the second classification is based on the substrate, right? So whether they can act on the single strand nucleic acid, they are called as single strand specific exonucleases. And the second class is even they can act on the double strand nucleic acid. They are double strand specific exonucleases. Now we will see some specific exonucleases and their action. E.Coli Exonuclease III initiates action from the 3' end of neck, and it is a double-strand DNA specific nuclease. Exonuclease 3. Here you see this is a double-strand DNA, and this is a. It says that it starts its activity from 3' end, 3' end, right? We will see that, and also it is a double-strand specific nuclease, right? Let us see the activity, right? So before you see here, this is a 3' to 5' and 3' to 5', right? Here 5' and 3'. So this exonuclease from the E. coli, let's say exonuclease 3, acts from the 3' to 5' direction only. So this is what you can see again. It is acting from 3' to 5' direction and cleaving that into mononucleotides. You see, again, right. The second one is exonuclease V, initiates initiates action from 3 prime or 5 prime direction. It's not specific. It can initiate its activity from 3 prime or 5 prime direction. Right? You see? Yes. So exonuclease 5 initiates the activity from 3 prime to 5 prime direction, or 3 prime or 5 prime direction. This one you see, the first one, it can act either side, right? So this is exonuclease 5. Then the next one is Exonuclease VII. It's a single strand DNA specific nuclease. Again, this also initiates the activity from 3 prime to 5 prime and 5 prime to 3 prime. No specificity. It can do the both ways. That's a one orientation. This was one more orientation, resulted into the mononucleotides. Okay. Then we will see some of the famous well-studied polymerases with the exonuclease activity. So you all know E.coli DNA Polymerase, which is having the two exonuclease activities. That's the 5 prime to 3 prime exonuclease activity and 3 prime to 5 prime exonuclease activity. Both are required for replication as well as repair in the physiology. And the second one is Taq DNA Polymerase is the most famous enzyme because it is used in the PCR, and that is thermostable DNA polymerase. It is having only 5 prime to 3 prime exonuclease activity, right? And third one is T7 DNA polymerase, which is having single strand and double strand 3 prime to 5 prime exonuclease activity. So these are the some of the polymerases which are having the exonuclease activity. Then we will move to the applications. So exonuclease 3 used in the sided mutagenesis, and also exonuclease 3 used in the preparation of single strand DNA for sequencing, and exonuclease 5 used in the plasmid preparation to degrade the linear genomic DNA, because it can degrade the linear DNA. When you are isolating the plasmid, genomic DNA will be in the linear form. To degrade that, you can use the exonuclease treatment. So hope you all like this lecture. So try to subscribe my channel for regular updates. And if at all you like it, share with your friends, and try to give thumbs up if you at all you like it, and try to write to me if you have any concerns. Thank you all for listening. See you again with the one more video. Until then, bye bye.